CRISPR-Cas technology. It is the 23rd talk of a series targeting to promote agrobiotechnology and enhance understanding of the potential applications of agricultural products. We hope the series will inspire international scholars, researchers, farmers, and business in the agricultural field, as well as the interested public. First, let me introduce our speaker today, Dr. Yiping Qi, who received a bachelor's degree in microbiology from Nankai Universities and a master's degree in biochemistry and molecular biology from Shanghai Jiao Tong University, and his PhD in plant biology from University of Minnesota, Twin Cities. From 2009 to 2013, he conducted postdoctoral research on plant genome engineering with Singvinger and talent at Dr. Dan Walter's lab. Dr. Qi started his own lab in 2013 at East Carolina University in North Carolina. He is currently an associate professor at University of Maryland, College Park. His lab has developed multiple CRISPR systems for plant genome editing and transcriptome regulation. His recent recognitions include NSF Plant Genome Early Career Award, FFAR New Innovator in Food and Agriculture Research Award, SIVB Young Scientist Award, UMD AGNR Faculty Research Award, and University of Maryland Invention of the Year in Life Science category. Genome editing technology based on CRISPR-Cas systems has revolutionized plant research and agriculture. In this talk, Dr. Qi will first provide a general overview of the landscape of the cutting-edge CRISPR genome editing tools, including CRISPR-Cas9, CRISPR-Cas12a, CRISPR-Cas12b, C to T based editors, A to G based editors, and prime editors. Then he will present a few case studies for the application of these tools in different plant species, including rice, maize, wheat, carrot, tomato, and poplar. Next, he will introduce highly efficient multiplexed genome editing systems based on CRISPR-Cas9 and Cas12a. Their powerful application will be showcased in rice, such as simultaneous editing of multiple crop traits, targeted protein evolution for herbicide resistance, and creation of quantitative traits by editing cis-regulatory elements. Finally, he will talk about an exciting gene activation technology, CRISPR-At 3.0, and its use for accelerating crop breeding and metabolic engineering. Collectively, these CRISPR technologies will greatly aid the development of high-yield, nutritious, and resilient crops to feed the growing population worldwide. Without further ado, let's welcome Professor Qi to the stage. Professor Qi, please.
Well, thank you very much, Dr. Lin, for your kind introduction. Wow, I was shocked that I provided you such a comprehensive abstract. I'm not sure I can deliver so many topics that you just mentioned, but let's see. The idea is really introduce genome editing tools to the audience. I suppose now you can all see my slide. First one here. Okay, so the title I give here is about cutting-edge crop breeding with CRISPR-Cas genome editing technologies. You all know CRISPR. So my talk here, really, I would rather give you a rather simple, maybe a brief overview of CRISPR, a little bit of history of CRISPR-Cas, and then maybe a little bit about technology landscape at this point. But just to confess, I don't think I have time to go through everything, as you may know why, because CRISPR has been developed so fast, and there's so much to learn, and I will direct you some resources to learn some of the technology to follow up. And rather, I will spend a little bit more time later to give you some kind of a deep dive into one or two technologies that my lab have been developing and has still ongoing, but we find very interesting results. I will share with you the most recent research. So that's really the point of this talk. So, all right. So what is the goal of my research? So my lab really wanna sort of democratize genome editing technologies for all the crops. So the goal is to make sure everybody, scientists, students, researchers, breeders, can really smoothly harness this powerful technology to apply for basic research as well as for translation research, such as breeding. So what is genome editing? So genome editing actually typically start with a sequence specific nucleus or sequence direct nucleus that enable. So these nucleus, they just function as a molecular scissor. So they will go to the genome, recognize the DNA sequence of interest, make DNA double-strand break. DNA double-strand break is sort of lethal for the cell, and then it has to be repaired by either non-homologous end-joining pathway or homologous recombination pathway. Sometimes we call that homology direct repair pathway. So either way, so the DNA can be repaired, and the NHEJ pathway is very efficient. And for the most part, it can repair broken ends very precisely. But because the precise repair will still render the sequence being targeted by this so-called sequence-specific nucleus, and then there's going to be many rounds of repair, and eventually the repair machinery may be tired and then making error. That is lead to targeted mutagenesis. We can knock out our gene of interest because you typically introduce small deletion and occasionally insertions at the targeted site. So if you introduce more than one DNA double-strand break, you have another scissor cutting here, and you would also delete the chromosome in between, or you can invert that, then ligate it back. So you can result in large chromosome deletion and inversion. Not to say if you have another scissor cutting another chromosome, you can also have potentially chromosome translocation. All these events were achieved by NHEJ. So HDR, if we provide a donor, which is kind of indicating green here, so you have a homology arm at the left and the right side. So this kind of for the system to sync this DNA is like a sister chromatids or homologous chromosome, so that you can repair without error. And this allows us to introduce specifically the DNA sequence, again in green, of interest. It can be some kind of gene replacement, small deletions, or insertions of DNA. Unfortunately, this pathway, the homologous recombination pathway, is being really low efficiency of human cells as well as in plant cells. It's only active during certain cell cycle, such as DNA replication stage. And plus, we have to provide a donor concurrently with DNA double-strand break. So it is really tricky to achieve high efficiency HR. And that's kind of a little bit unfortunate, because if you can achieve HR efficiently, you really don't need this NHJ pathway. But because HDR is highly inefficient, and most of the application we heard in the early years or until now for CRISPR-Cas9, for example, are still based NHEJ just for gene knockout, for example. So this gives you a little overview. I just want to emphasize whatever we have achieved for genome editing is actually through DNA repair, which we may not really necessarily talk about. But this is really, you need a DNA repair to finish the game. So, all right. The focus, of course, is so-called sequence-specific or sequence-direct nucleus here, because that's another name. I will come back. So in the early days, I have worked on zinc finger nucleus and a talon. So these are sequence-specific nucleus that need to be through protein engineering. So that is really not easy, not readily accessible for everybody to use. Even though I was very happy at the time, really excited working on these tools, because I feel that's really the future, right? Because this really allows us to modify DNA or gene of interest specifically. And luckily, in 2012, CRISPR-Cas was reported as an in vitro DNA targeting RNA-guided protein, sort of RNA complex nucleus. So that has really changed the whole landscape of genome editing, because just 1 year later, in 2013, CRISPR-Cas9 already become proven thing for genome editing in human cells as well as in plants. So ever since, there'll be a lot of CRISPR system being reported, such as this CRISPR CPF1, later called a Cas12a system, reported in 2015. So 1 year later or 2 years later, there's a lot of reports in plants, including my lab. So there's a lot of excitement, development, exciting in this field. So as we all know, that That's kind of why just really 8 years after its discovery, Nobel Prize in chemistry was really given to Dr. Champantel and Doudna for their invention of developing a tool for genome editing. So my lab has been since my lab my own lab is in 2013, almost 9 years now. So we've been always working on all kinds of CRISPR genome editing tools, such as Cas9, Cas12a, and Cas12b. For example, earlier we published CRISPR CPF1 for genome editing and also transcription repression. And I'm come back later why we can do transcription regulation with CRISPR. So we also have CRISPR-Cas12b to enable efficient plant genome editing, gene activation, and also gene repression. And these Cas12a or Cas12b systems are unique in the sense they can recognize AT-rich genome sequence, while Cas9 has been well known to recognize a GC-rich DNA sequence. And that's why later on we also developed, along with others, with a sort of a SPRY, which is a timeless Cas9 variant, which kind of basically overcome the bottleneck of Cas9 that only can target the double G or GC-rich region. Now this SPRY can literally targeting anywhere in the genome, although efficiency may vary. But nevertheless, these are all the tools will be very useful, allow researchers to target their gene of interest for genome editing. So there's more tools other than just those sequence specific or sequence direct nucleus, right? Those are just like scissor, can only make DNA double strand break. Additional tools have been developed since 2016. The first kind is called a base editor. So there are cytosine base editor, CBE, and also adenine base editor, ABE. So for illustration here, I just show you a cartoon of CBE. So where you would fuse, in this case, a cytidine deaminase to the N terminal of Cas9. And this Cas9 is a sort of modified Cas9 where this D10A mutation was introduced here. And because of the mutation, it cannot cut the non-targeting strand. It will only, it will still compress with guide RNA and targeting your DNA of interest and make only a single strand of DNA double strand break, DNA break here, which is cutting the targeting strand. However, because you have this cytidine deaminase, so it will actually convert the cytosine to uracil, C2U, because as we know, uracil U is actually an RNA nucleotide, not a DNA, right? So that is not recognized as a DNA nucleotide. It has to be removed by base excision repair pathway. And however, if we can fuse 1 or 2 copy of uracil glycosylation inhibitor, UGI, which can inhibit this sort of removal of uracil, and then this the uracil will be there, the U still there. So then DNA repair pathway or DNA replication will consider the U as a T. So then you can eventually achieve C2T base change. That's why it's called a base editor. So similarly, if you imagine you fuse a adenine deaminase at one end of this Cas9 nickase, you can actually achieve adenine base editing, ABE. So right now, these both technology, CBE and ABE, are already very mature, being widely used in plant genome editing. So if you haven't used them, just pay some attention. They can allow us to achieve specific C2T and A2G in a typically small window, like 4 to 8 nucleotide window. Another very exciting tool is called a base editor. So base editor is also utilizing a Cas9 nickase. And in this case, the nickase is actually nicking this, is targeting this non-targeting strand, which should be here. So in this cartoon here. And then what's really interesting is this guide RNA is modified. It has extended a 3' end. It has a sort of primer site. It has also the reverse transcription template. And all these allow us to introduce specific precise modification, because now we have a, let's say, very high efficient sort of reverse transcriptase. Okay, we typically use for RT-PCR. This reverse transcriptase can reverse transcribe RNA, which is coded by our guide RNA at this end, and especially this red part, and to DNA here. So basically, you just extended the DNA, and then this DNA will ligate back. So this allows us to introduce a few nucleotide of change, not very long, but enough to make like large deletion as large as 50 base pair and also insertion maybe to a few dozen base pairs. So this is a prime editor. So prime editor is a very nice complementary tool to this base editors, because base editor can only make C to T, A to G, while prime editor, you can make literally all the 12 different nucleotide switch sort of changes, and also making small insertion and deletions. The only thing prime editor, there are two limitations of prime editor. One limitation is prime editing can only make small modification. You cannot really insert very, very large DNA sequence. So that's one limitation. Second is right now, prime editor are overall still of low efficiency. There's a lot of researchers are actually actively working on improving prime editing. So as you know, there's a lot of tool has been developed. So to really give to help other people in the plant biology field to kind of keep up with the technology, we constantly sort of summarize the most recent breakthroughs and their applications as review papers. So there are many reviews, and even my lab wrote many reviews just for that sake of that reason. So just two reviews I want to introduce to the audience here. So one is like 2019, another one is relatively recent 2021. The recent one already kind of focused on base editors and a prime editor. So interested readers, really want to have a good overview of the technology of CRISPR, and I encourage you to look at these two review papers. So one thing people are really excited about CRISPR or genome editing tool is it is really a kind of a new breeding technology, and people consider they are different from a GMO or a GM or that created GMO, and which is kind of controversial across global, right? So we know not many countries actually can grow GMO without regulation. So when CRISPR become a tool, so people realize, well, it can make some deletion insertions, but naturally or mutation breeding kind of introduce those kind of mutations, which are considered as conventional breeding, right? So that is why we consider CRISPR as an alternative technology or better technology, sort of compared to conventional breeding tools. But we have to understand, or we have to learn how precise these tools are. Is there any risk or safety concern about using these tools, right? So from a technology point of view, we want to know whether there's any off-targeting mutations introduced while you are making your own targeting mutations, right? So what about other sequence in the genome-wide? So this is really the question that quite a few years back, we tried to investigate, which is partly funded by USDA NIFA, because for the regulatory agencies, they want to know how safe these new technology are to modify the genome of crops. So for instance, I've been involved in two sort of collaborative research simultaneously. One is really with collaboration with Dr. Ken Wang at Iowa State University, and also Kis Jong at Harvard Medical School. And what we have done is we really try some in vitro assay to assess the guide RNA specificity, and then get a targeted amplicon sequencing of many potential off-target sites in maize, just to assess CRISPR-Cas9 and CRISPR-Cas12a, how safe these tools are. Is there any off-target mutation at the predicted off-target sites? So this is as much as we can do. We just cannot do at the time of whole genome sequencing, in-depth whole genome sequencing with maize, because it's very repetitive genome. It's very hard to figure out. We have really covered every ground. So, but we can do those combining in vitro and also targeted DNA sequencing to evaluate this aspect. So in a second study also happened concurrently is I collaborate with Dr. Yong Zhang and Tao Zhang in mainland China. So this is really, we picked a model crop, which is rice. Luckily, rice is not unlike a maize, which has 3 sort of gigabase genome. Rice is only about a little bit over 400 million base pair. The genome is not that redundant, don't have so many transposons, for example, and then it's well sort of annotated genome. So we can really do whole genome sequencing to reveal any potential off-targeting effect. So long story short, because the paper being published quite a few years back, so we actually found no evidence that's actually sort of guide RNA, either guide RNA dependent, a little bit evidence of guide RNA dependent, but if you design guide RNA very well, just like you design PCR primer, and you can avoid this off-targeting. And then we didn't find any guide RNA independent DNA cleavage result mutation by either Cas9 or Cas12a. So overall, we conclude these top two CRISPR technology are really precise genome editing technology. So people can feel free or safe to use them as long as you design your guide sort of very specifically, such as your guide RNA of 20 nucleotides, let's say, will not have high homology to elsewhere in the genome other than the target sequence you are interested. So that is a very good contribution to the regulatory agencies. For instance, in 2020, more than 2 years ago now, so USDA, APHIS at the United States here kind of made formalized a sort of a regulatory law which could secure. This law will turn many CRISPR-based genome editing, such as make small insertion and deletion, and if you use base editor, only introduce one nucleotide chain in the genome, such as make an amino acid change in the protein of your sequence, right? So they will not consider those as a GMO to be regulated. So as long as those plants don't carry any CRISPR-Cas transgene. So this important ruling, first of its kind probably in the world, is really kind of revolutionizing, I think, the landscape of regulation, I think. So, and really nice to see these two papers I just mentioned, looking at off-targeting effect of maize and also off-targeting effect in rice for these tools really being cited by this important ruling and to consider technology as safe. So as you can see, then, a year later, we learned the news like in Japan, there's actually first the genome editing, gene edited tomato goes to shelf, right? Making some splash in the news. And so far, so good. A lot of, countries follow suit, like we know Australia also have a relative liberated regulation, just like US, Japan, Australia. I think also a few South American countries are like that now. So a lot of countries can really, have genome editing go to the market because of the very friendly regulation. So India is also seem to be follow suit as well. So this is one of the, I think, the letter I received from a collaborator. I don't know when, not too long ago from India. Maybe half a year ago. So basically, the sort of SDN1, SDN2 sort of editing, which means sequence direct nuclease 1 type, which makes insertion deletion, and type 2 is making me some specific replacement of the genes, very small change. And these two they consider will be exempted from a regulation. So these two would not really considered as a GMO, basically. So that will be very exciting, right? Because India is a big country, and also there's a lot of agriculture research, germ plant development are ongoing. So you can imagine if everything going well, then India will catching up with applying these tools to breeding and making product in the field, hopefully. But why people are so excited rushing to use genome editing tool? Well, let me summarize that here a little bit. So here we look at a few matrix, and by comparing GMO or based through GM technology, just typical transgene, conventional transgene technology versus genome editing technology. So if you look at regulation I just mentioned, right? So genome editing is potentially light regulation and not regulated depending on what kind of editing outcome is. So not regulated in many countries, some countries already, I just showed you, while conventional GMO, even in the United States here, it's regulated, right? So even though we have a lot of GMO crop growing in the US, like soybean, corn, but they are regulated, they're heavily regulated. So in fact, currently only permitted in about 30 countries. The timeline to trade, and conventionally, GM technology takes more than, like, let's say a decade, like 13 years on average, while genome editing with some estimation is maybe only 5 years. You really, save a lot of time. And the cost to develop a new trait, this is very important. So for conventional GM, on average, it takes $130 million. It's almost like making a drug, for treating human disease, right? It's very expensive. While genome editing, by some estimation, only cost about $10 million or even cheaper, depending on which crop you are doing, which kind of edits you are doing. Okay, so it can vary, but overall much cheaper. And then available crop. This is again very, very important. And if you look at the GM technology, it's really only applied in large scale, okay, in the marketplace to corn, soybean, cotton, canola, and papaya. And papaya, as we know, because of the virus problem. Okay, so that's why almost you have to, we are in a desperation mode. We have to try some GM to really save the industry of papaya. So that is why papaya is enjoying that. But other than that, the corn, soybean, cotton, canola are really huge cash crops, such as North American here. That is why the big biotech company can afford $136 million per average per trait to engineer GM. I mean, because these are cash crops, you can potentially recover the revenue, right? So, but that's also why GM really didn't benefit many of the vegetable, many of the other, third world, right? So because it's too expensive, which result in monopoly of big companies like Monsanto. Monsanto now disappeared, and now you have Bayer, and also you have Syngenta. You have Pioneer, DuPont, but now you have Corteva, and you have BASF. These really top 3-4 major biotech companies are dominating a monopoly this GM world, right? This is not very good. This is not also considered being very good from public eyes, because they feel these are for profit. Big giant companies are control technology, must be evil. So that didn't really help promotion of very nice GM, I mean, transgene technology. Unfortunately, this is a mistake we kind of have to avoid when we promoting genome editing technology. And because genome editing is really, really fast, not even regulated, and relative, because of that reason, it's cheap to apply, develop crop. And now, because of that reason, now we can actually apply to many crops as long as we can deliver genome editing tools into those crop species. So there is really a brief overview to give audience here to understand why genome editing is really being promoted. The one big reason is the regulation is very light. It's almost equivalent to conventional breeding, and it's precise to make that process very fast, and because the reason make very, very cheap. And it's excitingly, we believe it can apply to many, many things like lettuce, carrots, apple, any kind of the agriculture important, a product. Everybody's interested. We can potentially use genome editing as long as you can deliver CRISPR, let's say, into the system. So this is my kind of summary with my first part. So about the overall landscape of the technology and overall landscape of the regulation, very brief. But now I'm gonna save the second half of my talk to give you some update on something you may not necessarily heard of, or you may not necessarily look into deeply, okay? So when we talk about CRISPR-Cas, we often think of genome editing, as I showed you, making modification at a DNA level. So even though I only mentioned about the nuclear DNA genome, but plastic genome and mitochondrial genome are also, we also have other tools for genome editing. I just don't have time to cover that. But we can also repurpose such tool to do other things like modify transcriptome to turn up or tune down a gene expression, or we can make modification in the histones or chromatin, like histone modification or DNA methylation. We can modify those epigenetic marks. We can develop epigenome modifier and change those epigenome marks. Eventually, that will also turn back to change gene expression. So ultimately, we have all kind of tool which allow us to do sort of transcription regulation of genes rather than make mutation at the DNA level. So I think this is also very potentially very, very powerful tool. So it's only become more discussed and developed in recent years. And my lab is always kind of one of the sort of front runner in that aspect. So before I jump into the topic, I just want to show you CRISPR-Cas is an amazing tool because it is a it's RNA guided DNA targeting system, right? And I already showed you the Cas9 nickers, depending on nicking which strand of DNA, and then it can help us to generate the base editors for base editing or the prime editors for prime editing. But that is nicking, right? If we further make another mutation so that it cannot even nick a DNA, it just becomes dead, deactivated Cas9. And then, interestingly, it can still compose guide RNA, okay, to bind the DNA, just cannot cut DNA, just cannot introduce double strand or single strand nicking at all, right? But it can bind. So now you literally converts CRISPR-Cas system to a homing system, which you can target that to a DNA of interest still, right? You just don't do genome editing. You just, but you can control gene expression For example, you can fuse sort of some transferrin effectors to Cas9 or the guide RNA targeting the protein and to control gene expression. And as I said, you can have epigenetic modifier to modify the chromatin, also result change of gene expression. Or we can really recruit a fluorescent protein like GFP to do in vivo chromosome DNA labeling to replace fish technology. And you can also sort of use this technology to pull down a chromosome, doing some chip kind of analysis to look at what's really being associated in this region of DNA in the chromatin, right? You can follow that with mass spectrometry. So you can do a lot of things really, right? So this is very, very powerful. I think that is another reason why CRISPR, that so many people has been published with CRISPR, there are so many discoveries made with CRISPR. And I didn't even show you here. These are all about manipulating the genome and also the transcriptome. But CRISPR can also be used as a diagnostic tools to detect pathogens such as SARS-CoV-2, which is called the pathogen for the COVID-19. So we can also use that to detect different kind of pathogen, which is another aspect my lab is also working on. I don't have time to talk about that, but I just want to give you some idea. CRISPR is really, really a versatile system. So from now on, I'm going to only focus on one aspect in addition to genome editing. That is CRISPR activation, CRISPR-A system. So CRISPR, as I showed you, if we make a deactivated Cas9, it can still bind to the DNA. In this case, we are targeting a promoter region of interest. And we're going to recruit some transcription activators, such as VP64, right? Then we can activate some gene expression. This is showing a model plant Agrobiotechnology. So these two genes can be activated to a few, up to 7-8 fold. And then we can recruit more such transcription activators. For instance, we can engineer the guide RNA with 2 additional RNA stem loop, and each of them will accommodating binding to a dimer of the MSCP protein, because MCP is kind of binding to the MS2 stem loop, this RNA binding protein interaction. And then we can fuse VP64 with MCP, and then you can see now we can recruit more VP64, and indeed that translates to higher activation potency. For the same gene, POP1, we can activate 40-fold rather than 4-fold here. And for this FIS2 gene, which is basically silenced, that we can activate to the 1,000 level higher compared to the sort of original level. So this is really CRISPR activation can work in plant. However, even though we have developed this CRISPR-A 1.0, CRISPR activation 2.0, we still realize there's more room for development. That is why relatively recently, my postdoc Changtian Pan developed this nice CRISPR activation 3.0 system, 3rd generation, which is already published about a year ago. And then this system, just briefly describe, this system is basically actually Changtian tried many different ways you can imagine to recruit transcription activators to the promoter region of the gene of our interest for gene activation. And this is this system here is that eventually he ended up with by all kind of comparison. So this system is based on a modified guide RNA 2.0 I already introduced you, which has 2 MS2 optimal. And then MS2, we have MCP, which will bind to MS2 as a dimer. So now imagine if we can fuse a SUNTAG, which means here is 10 repeats of GCN4 peptide, okay, fused to this MCP protein. And then we have another fusion protein, which is you have a transcription activator, and then fused to a SCFV single chain antibody, which will recognize this peptide. Okay, you have 10 peptide. Technically, you may have 10 antibody to have one-on-one relationship binding. Okay, so and in fact, in this case, we use a superfold GFP as a linker. So when you have a guide RNA designed to target the promoter interest, so this modified guide RNA will now recruit this SUNTAG, and because MCP bind to MS2 stem loop as dimer, so each stem will recruit 2 tail of SUNTAG, and literally you will have 4 SUNTAG repeats, and then each of the tail will recruit many copy of the activators. And Changtian actually compared many different transcription activators, has been shown working, so derived from plant pathogen, from you worked in human, worked in plants like VP64, as I showed you, all kind of activators he compared, but eventually he ended up this one. The activator is the 2 times the 10m repeats of tau activation domain originally from the tau effect protein from Desmonas pathogen. So this is the activator Chang Tian found is most potent. So he actually have to compare with all kind of tools already developed by us or by other researchers, including this DCAS SunTag system, with recruiting a specific activator different from ours, and also the TV system, just the multiple fusion of the tau activation domain and the VP64, and also this DCAS EV 2.1 system, which recruit activator of EDAL by Cas9 fusion, and also VPR activator by guide RNA modification. So as you can see, we actually compared these. As you can see, these are the 3 other versions, and our system here, they either use a U3 promoter or the ubiquitin promoter to drive the guide RNA, and we always can achieve much higher efficiency for gene activation with like over 100, about 100 and over 100 fold activation of your targeted gene, which is 2 here, GW7 and ER1, compared to these activator. They are working, but our system is much, much stronger. So this result is actually derived from the protoplast of rice. So people may question, okay, so you showed transgenic activation. What about that only at the mRNA level? What about protein? Will that really translate to protein? And then you have a phenotype, right? So then to do that, we basically fuse our mCherry reporter to 2 very weak promoter, which if you drive that mCherry with weak promoter, you cannot see expression at all under a microscope, and in the protoplast of rice again. But if we use our system, just using 1 single guide RNA to recruit many activators, you can see it light up cells very nicely, suggesting that can translate to really protein level highly. And then Chang Tian made this highly multiplexable, because the beauty of CRISPR-Cas is we can do multiplexed genome engineering, meaning you design multiple guide RNA to target multiple genes of your interest. In this case, it's multiple promoters for the genes of our interest to activate them, right? So we have the cloning system for you to do that very easily. And then to demonstrate some sort of proof of concept, we can do sort of metabolic engineering. And for instance, here we are targeting 6 enzyme pathway genes in the proanthocyanidin pathways. And you can see here in the protoplast, 5 out of 6 are being activated at different level, and in the transgenic callus, they can nearly all be activated, as well as in the transgenic seedlings, even though the fold change has been reduced. So you can really activate many genes at once with our CRISPR activation. So, but that is from the rice cells. What about the dicot? So in this case, we try to activate the FT and also TCL1 gene, just proof concept, and make a single tDNA vector with, we designed 2 guide RNA because we didn't have time to optimize guide RNA just to be safe. We kind of designed 2 guide RNA to target each promoter. As you can see, in this case, FT is being activated. We have early flowering. I'm going to come back for FT shortly. And then you can also see that because of that, the life cycle has been really reduced by 1 month, and from seed to seed with FT activation. And also activating the other gene will reduce the number of trichome. And indeed, this is what we found. But in short, the activation not only translate to protein level, but also can have a phenotype. So this really makes CRISPR activation as a powerful gain of function, right? CRISPR system is usually used for knocking out the genes, right? Making mutants. But it's really limited if you think about engineering a trait by mutating a gene. It's very hard, right? So it's actually much, much more powerful if you can activate the genes, have more of the same product of the genes to engineer crop. So that is why I think this is a very exciting tool. And then Chang Tian also showed we can design guide RNA for gene activation in tomatoes. So basically, this system works for across many plant species. And again, that's another idea. Think, okay, you can activating a pathway gene. I showed you proanthocyanidin pathway. What about the beta carotene pathway? So that we can engineer golden rice, right? So we have golden rice #1 and #2. After more than 20 or 20 years, the golden rice was just like really approved in Philippines, right? So we know that maybe a couple of years ago only. It takes so much time to get regulatory approval, which is unfortunate. But in our cases, we can propose, well, maybe we can because of the pathway of this beta carotene, which is a precursor for vitamin A, is inexist, right? Because it is like involving photosynthesis, and it's almost in all the cell type. It just matter whether we can, have its expression or not. So in this case, we basically designed 7 guide RNA to target the enzyme gene in this beta carotene biosynthesis pathway. As you can see, well, they can be activated to different level. So now we are investigating the new approach of using CRISPR activation to engineer sort of a golden rice. So this is another interesting project ongoing in the lab. So as I mentioned, activation is very useful. That's why, in fact, Kevin Fota engaged a sort of interview with me in this podcast, talking about the potential and aspect and the usefulness of this gene activation rather than gene knockout with CRISPR. So the last story is continuation from the CRISPR-Cas 3.0. Even though it is really a nice gain of function tool, but it's just really a tool that adds to the sort of a CRISPR toolbox, just like you have many tools in the Swiss Army knife toolbox, right? So almost everyone have one in your family. So yeah, we use such tool, but we really, depending on the needs, we only use them 1 tool at a time, right? You barely think about I can use 2 at a time. No, it's very hard, right? But we already have genome editing tool. Now we have gene activation tool, right? Because one is modify the genome, another one is modify the transcriptome. So it will be nice, we can combine them to do simultaneous modification, just like the smartphone we are having, everybody have one. And you have more than one app is running constantly, even though you're not using your smartphone, right? So I think having more apps, more tools can work simultaneously. It is very powerful. So we just don't imagine it typically to think about what's the use of this kind of tools when you do them together. But once you have them, I think it will be really promote people's imagination and creativity to do a lot of beautiful things, which I highly believe. And at least I have some idea why these tools will be very useful if we develop that. So I call that CRISPR combo, which is a combination of genome editing and gene activation together. But first of all, we want to know how to develop CRISPR combo efficiently or useful system. So we just evaluate a few parameters here. We truncated the guide RNA, which is usually 20 nucleotide, to 14. So as you can see, when you make nucleotide shorter, this targeting guide RNA, and then down to 17, it can still make mutation. We can detect it with this result here. But if you make short guide, there's no mutation, which means it cannot cut the DNA anymore. But you may wonder whether maybe it's not even binding to DNA. Well, we'll see here. So we also use a truncated guide RNA for gene activation to bind this promoter of GW7. So as you can see, actually this 20 to 17 guide, they activate the gene, but they are doing a good job here, right? It's just not really highly activation here. But if you use shorter guide, okay, so they actually can activate gene highly. So which actually suggests that these shorter guide indeed allow CRISPR-Cas to bind the targeted DNA. They just cannot unleash the nucleus activity to make DNA breaks. So which is nice. Now but you ask why your longer guide didn't result in highly efficient gene activation. Well, because this longer guide is resulting genome editing, making mutation at where you are targeting. And once you make a mutation, the guide cannot bind anymore. Of course, that result inefficient activation. Makes sense, right? This is from population of protoplast. So with this result, we can easily conclude that you have guide RNA more than 17 or equal to more than 17, you can do genome editing. Use guide or protospacer length is about 16 or shorter, like about up to, let's say, 14, you can do gene activation. It's that simple. Basically, we can truncate the guide RNA to, let's say, 15 or 14. Now you are literally convert a CRISPR-Cas system to a DNA binding system, even though the Cas9 is actually a Cas9 nickase. But by using shorter guide RNA, we literally convert the system to a dead Cas almost. So because of that reason, now we can engineer CRISPR combo. I'll show you how. In the CRISPR combo system, you will express the Cas9 nucleus, and you will have our activation component for gene activation I introduced you earlier. And you will simultaneously express 2 kinds of guide RNA. Okay, one is guide RNA scaffold 1.0, typical guide RNA we use for genome editing, and which is paired with 20 nucleotide protospacer or guide, which will result in targeting your DNA of interest. and knocking out your gene, let's say make a double-strand break, right? And then the second guide is called guide RNA 2.0, which has stem loop. The stem loop will help recruit these activators, right? So then you can target a promoter to activate your gene of interest. And because we only pair these modified guide RNA 2.0 with a shorter target guide, 15 nucleotide. So even you can target a bind to your promoter, but it cannot really cut the DNA because it's used a shorter guide. So this allows you to do genome editing at one gene, and simultaneously in the same cell or same plant, you can activate another gene in the same time. So this is really the principle of how CRISPR combo can work. Of course, we want to show whether it really can work. And indeed, we compare that for gene activation and this, the CRISPR 3.0 that one, and the Cas9 nuclease-based activation 3.0, which is CRISPR combo literally. So we can have same level activation for genome editing. This Cas9 nuclease version-based activation system, again, combo, is as efficient as the naked Cas9 for genome editing at different kind of PAM sites in the rice protoplasts. And same thing observed in tomato for gene activation, gene editing. Not only that, even for base editing. So as I earlier introduced you, if we fuse a deaminase to Cas9 nickase, we can achieve base editing here, right? So we can do base editing. And then we also test the base editor, derive the combo system. And we found, interestingly, the CBE-based combo system can activate a gene as good or even better than the typical activation system. But however, ABE can only activate it to a relatively lower fold activation potency here. So, which is interesting, we're still investigating why, but nevertheless, the CBE system is a highly efficient combo system to make a C2T base change in the genome, while we can simultaneously activating another gene. So base editing is highly efficient. The CBE AB is as good as a typical CBE AB system, as you can see here in rice. Same observation, because CBE only worked well, much better than ABE in rice. So in tomato, we just focus on CBE, and indeed, CBE works very well for base editing, just like a base editor here, and also for gene activation. So again, in principle, and also as our results showed, it worked as we designed. But the question next is, okay, why you design that? You just make complicated tools. What is really meaning? What's the meaningful application of that, right? So I'm going to show you a couple of, I think, very meaningful application. Okay, so this is the CRISPR combo. We're going to be using that to overcome two of the major bottleneck or limitation of genome editing application as a breeding technology in crops. Okay, so one thing for crop breeding we think about is actually breeding cycle. So that is why when we see CRISPR-Cas, so many of them are used in annual crop because you can at least breed them like once. Many people like to go to Hainan Island of China to do rice breeding because you can have like more than one season, right? So that's really just you can gain another cycle of breeding. Really, that is why, So really to shorten the breeding cycle is ultimately very important to speed up the process of engineer next generation crops. And secondly is really tissue culture. So even though I showed you in the summary table earlier that, well, yeah, we hope CRISPR-Cas can be applied to benefit many crops, many some niche crops or some small vegetables. But the question is that you have to be able to deliver the genome editing technology, before you can achieve that, right? So which means we have to find a way to really get the plant tissue culture regeneration working. Otherwise, you just cannot generate the genome editing crop. So these are really two major bottlenecks, in terms of application of technology. So again, we know as a speed breeding, we can control the environment to make really breeding faster, like using longer daylight and also some fancy kind of light system to make it just faster growth cycle faster. And then you can really making the development stage from 5 to 10 years become 1 to 2 years, right? Just grow them in the greenhouse or growth chamber rather than in the field. So this is really the idea of speed breeding we all know, but we can actually use CRISPR combo to achieve a sort of genetic speed breeding. Okay, this is back to the earlier data I showed you with FT activation. I don't have to overly spend much time because now using combo, we can designate a guide RNA to activate a gene is FT, right? So then you have the early flowering, even in the petri dish, you can see they start flowering much faster than, conventionally you're using CRISPR-Cas, nothing happened, right, at this stage. And this is Cas9 simultaneously editing 2 genes here. And the same thing, we have combo simultaneous editing 2 genes, but on top of that, we have another guide RNA for activating the FT using a shorter guide. So it won't edit, but only bind to target promoter to activate the gene. What's really nice is both gene activation and genome editing are achieved by a single Cas protein, as I showed you, the combo through a single Cas nucleus protein, meaning the function or the outcome of genome editing and gene activation can report each other, vice versa, right? So this is very, very useful. So for instance, for these earlier flowering plants, the genome editing is highly efficient because these early flowering plants, meaning they are being, having high level of CRISPR component for gene activation, meaning you have high expression of Cas9. Of course, that will translate to high genome editing efficiency. So this become a selection surrogate reporter system, which is kind of nice byproduct. We can also do a CD, cytidine deaminase, and similarly, you can have highly early flowering plants have small dynamic range of, you can look at the individual transgenic plants, which meaning the robust genome editing is more robust with this by picking the plants with early flowering. And then the next generation from T1 to T2, we can pick plants that are actually not early flowering, they're just normal. Because those early flowering just means they are transgenic, they're GMO, because they have the CRISPR-Cas transgene, they're activating the FT, right? So if we only focus on the normal plants, and indeed 90% or more chance these normal plants, if you genotype that, it is transgene free. That it is, they segregate out the transgene. So just looking at the phenotype, instead of doing all the molecular analysis, now you can actually understand, pick the plants which are not transgenic, and then you can genotype the plants and whether it's really contain edits or not. It saves you a lot of work. And then you go to T2 and you genotype the plants. We find some of them have edits, and indeed at the T3 generation we grow them up, the seeds, and all these modified plants are resistant to the herbicide. Because in this case we make one specific amino acid change, the plant will become resistant to the herbicide, and that is really expected result. So this is just quick summary of 3 advantages of so-called genetic speed breeding approach, where we will basically have sort of early flowering, which is really a save your seed to seed life cycle by 1 month compared to conventionally you do that. Number 2 is because early flowering, you can just pick the early flowering plants, which you are enriching the events of genome editing rather than randomly picking plants here like it's conventionally. And finally, we in the next generation, we only pick the plants which looks normal, which means they are tDNA free for the most part. And then you can only genotype them to look at the edits. Then you will immediately get a genome edited crop of plants without the transgene. So that kind of plant will not be regulated in some countries, as I showed you, even though this is really proved concept using Agrobiotechnology, but you can imagine you can really readily apply it to all the annual crops. It's the same idea. So I kind of really don't have much time. I'm just going to quickly go through a second application, which is overcome the tissue culture and regeneration. So one example is about poplar, which is very important, the bioenergy, biomaterial tree my lab is also working on. So in this case, instead of activating the FT for promoting early flowering, we activate the so-called morphogenic factor genes like WUSHO here. So you can see activating WUSHO really promote rapid looting and shooting the plants grow much, much healthier, faster than the typical CRISPR-Cas construct. You just have guide RNA for gene editing. And we, in this case, we're still editing this gene, the 4CL1 involved in lignin biosynthesis gene, but we also activating WUSHO. And then you can see the days to loot reduced to half, and then looting rate enhanced from 50% to 100%. And if you look at individual transgenic poplars, the gene of WUSHO indeed are being activated at different level. So the genome editing efficiency is compatible, and you can look at the sort of genotype here, a few example lines. And importantly, if we can pick this this high expression line number 4 here, and then we do a perio cutting, and then to put them into media, as you can see, 100% of them sort of generate loots quickly, while normally with Cas9 only 50% you generate loot. So very sufficient and robust sort of propagation looting system by activating WUSHO. And then you can also achieve a similar result with activating another morphogenic factor, WOX11 gene. Similarly, individual poplar, if you look at 10 plants here, they all have this gene activated at different level, 100 level easily. And genome editing is as good as typical CRISPR system. And some of the genotype of the edits here you can find. And again, this show you 3 lines, number 2, number 3, number 6, because they're high expression lines, and we follow them. As you can see, they really have enhanced sort of callus formation compared to wild type control, and then the shooting is more robust compared to control, and we can quantify that. And in fact, activating moisture indeed promote this tissue culture process. And also for the looting, so you can take these high expression line number 2, 3, 6, and do some cutting, and then you can see they form root much more robustly, and the plants grow actually much larger because that reason. Again, days to root also reduce a lot. So this is again just simultaneously plugging another guide RNA, which allow you to modify the transcriptome. You can modify the plant phenotype along the genome editing outcome. The final example I give you is move from a poplar, which is a perennial dicot tree, to a rice, which is an annual monocot crop. Okay, so in this case, we target rice because rice is so important for let's say for Asia, like China, India. We are relying on rice so much. Everybody is eating rice every day. So this is very important. So what we try to do here is try to activate another gene. It's also a morphogenic factor called the baby boom 1 gene. Why? Because this gene has been implicated or involved in embryo formation. Very critical, function from a single cell, from an embryo become a whole plant, as we know. So through control different hormone pathways. So we try to activating baby boom 1 because as for proof of concept, we also sort of modify these 2 genes here, GW2 and also GN1, because this will make the seed size larger, and another one is make more seeds in the panicle upon genome editing. So basically, we can sort of enhancing the yield by modifying these 2 genes simultaneously. But on top of that, we activated this baby boom 1 gene here. So, and indeed, as you can see, with baby boom 1 gene, and we actually compared to typical CRISPR-Cas genome editing construct. Okay, you can see how many more of a seedling we can generated from these callus versus typically this is how much you regenerate for petri dish, a lot more. And that's why this is being summarized in the statistics. And then basically again quantified that there's more transgenic plants per callus, okay, with activating a baby boom 1 compared to normal CRISPR-Cas construct. More importantly, looks like the genome editing efficiency is improved, as you kind of understand why, because selecting of this plants, really you are selecting the high expression lines. That's why genome editing is also highly enriched in these lines. And finally, I want to show you very exciting data actually. So when Changtian showed me data, I said, why not since you're activating a gene to enable tissue culture, and we know when we do tissue culture, you have auxin and the cytokinin on the petri dish on the media to sort of use hormone to reprogram the transcriptome of plants and then reprogram that to regenerate. Since now you have, you can directly enable gene activation of baby boom 1. Why don't you just remove all the hormone from media, right? Because you may not need that. That is indeed the case. So Changtian we removed all the hormone, of course. Without hormone, typical CRISPR-Cas construct, you don't have anything. Everything eventually die. But just with activating 1 gene here, with baby boom 1 gene, you can see these callus can still form and grow and become eventually regenerate. And amazingly, I can tell you, they actually enrich high efficiency genome editing. This guide RNA for this gene, GN1A, is almost very, very poor, which typically results 5% editing or 10% editing, but now it's almost 100% editing. So this has really become this hormone-free CRISPR combo-based genome editing system it's by itself become a powerful genome editing system, representing a very, very efficient genome editing system compared to conventional one. So that is what I have. This is a summary slide just to say we can, with hormone, you can, by activating baby boom 1, you can achieve a faster, robust regeneration with higher genome editing efficiency. And in an extreme case, even without hormone, we can actually achieve a plant regeneration, highly enriching genome editing event. So this is highly, highly powerful, useful technology. And again, with time and with, so I only introduce you 2 application of combo system. One is the genetic-based spread breeding. Another one is to enhance the regeneration and plant tissue culture in a tree, as well as in the monocot rice. But they can also be applied to other systems like metabolic engineering. I showed you we can simultaneously activate the pro-anthocyanidin pathway genes and beta-carotene genes, right? But what if you can also, using CRISPR-Cas to knock out other competing pathway, right? You turn up some gene while you destroy other competing pathway gene. You can do very complicated metabolic engineering. And also you can imagine we may be able to do some combination of screen while you sort of have a gene gain of function, and also you have simultaneous loss of function by CRISPR knockout happening at the same time. So I think these are all potential applications. And with that, I want to thank many people. Actually, I didn't really have room to show many of the collaborators because many of the research I'm doing was really through collaboration, international collaborations as well. And then all the funding agencies that support my research over the years after I moved to UMD since 2017. And most importantly, all the people in my lab who do the hard work. So thank you all. I'll be happy to answer any questions.
Yeah, thank you very much. I think it's a very exciting talk, open up a lot of possibility for crop improvement. So now we entertain questions from the audience. Please make use of this opportunity to learn more. But then I start first, right? So then we try to understand the combo is that you can do multiple things in the same construct, right? So
yes,
yeah, so you can use one to activate the promoter region by bringing in some activation factors, or at the same time you can add some targeted regions, right?
mm-hmm.
And maybe I'm not. I want to clarify one thing. So your system is very much error-free, right? So the off-target problem seems not to make major problem here. Maybe I missed the reason. So why your system is so precise that I think that is a major problem that people worry is the precision of the target. And that's lots of environmental activists believe that when you have off target, you create something that's unpredictable. So could you kind of briefly explain how your system are so accurate that you can prevent this off target problem?
Oh yeah, so this is actually mostly about the CRISPR-Cas system themselves, about how precise. As I showed earlier, so based on our kind of data and result, CRISPR-Cas is literally very precise. You just so if you design a 20 nucleotide guide, which we typically do, and you just make sure the second, if you do a blast of genome, okay, so the second potential or off-targeting sites or whatever homology site to your guide RNA has at least 2 mismatch nucleotide, okay, in the 20 nucleotide sequence. You are pretty much very safe to say you won't have much off-targeting at all, because even the one has 2 nucleotide mismatch depending on the location though, but they usually don't get edited easily at all. So if you can avoid those, it's just about the design, like you design PCR primer, right? So if you design, a very promiscuous primer, of course it will amplify something random, So, but if you design very precisely, then, yeah, because this is really, the off-targeting is really mostly based on the RNA DNA sort of hybridization, specific hybridization. But one caveat though is actually when you expand the genome editing to base editing, let's say, and then the base editor, because base editor are all derived using those single-strand DNA binding deaminase. So they will give you a potential chance of off-targeting, which my lab already published 2 papers in base editing system off-targeting effect. We have another one preparing now. So this, I think, second paper may not be fully published at the Biotech Journal, but it's already posted at bioRxiv many months ago. So my point is, when you try different base editing systems, they more or less sometimes you're unlucky, you'll give you a little background mutation because the plant's expressing high level of the nucleus. Okay, so but if you compare those background mutation, we can predict almost some of the background mutation, what kind of, but sometimes you cannot predict random, right? So, but if you compare, it's really ironic. If you compare those mutation compared to the mutation which derive from tissue culture, we call somal clonal variation. So somal clonal variation is anywhere from 100 to 200, per generation for rice, and it can be 300, 400, 500 for tomato during tissue culture. So you always have mutation introduced. That's why somal clonal variation tissue culture become a breeding technology. You know that because it can make mutations randomly. So people use that as a mutation breeding, just do tissue culture, and then mutation happens, then you eventually may have lucky to have a trait mutating a gene. So, but that's considered as a conventional breeding, right? Not regulated. So my point is that even you have off-targeting, what we have found doing some mass calculation, we barely find off-targeting mutation with base editing in this case is with far more mutation produced, compared to the somoclonal variation derived from tissue culture. So from regulatory perspective, you can argue, why if conventional breeding need not to be regulated, not regulated, why you wouldn't regulate us, so you can do all kind of argument on that. But to me, if we are doing genetics, right, if you're designing a guide RNA to target a gene and you have a phenotype, you of course have to worry about, oh, is that due to off-targeting, even though the off-targeting potential is very low, right? But you can easily do that. The beauty is even you design a single guide RNA to target your gene, and it can make a bunch of different variety of deletions, 3 base pair deletions, 5 base pair deletion, 1 base pair insertion. You can have easily like a dozen allele of your mutants generated. And then you can look at whether all these allele have shared the same phenotype or not, right? If they all have same phenotype genetically, that is very convincing to say, the mutant phenotype is result from knocking out this gene, even without doing any complementary assay. So I think that's very powerful because you have so many alleles generated. So.
Yeah, so to the other side of the story, right? So some genome are polyploid, like tetraploid, even hexaploid. So will your system facilitate a rapid turn off of all copies of genes in the tetraploid, like our hexaploid system? Is it you can split it up?
I think so. I think the principle applies, right? But as I showed you, for the rice, we activating depending on how you use the combo system. In rice, we activated this OS baby boom 1 gene. In poplar, we tried 3 genes. 2 of them worked. I showed you. One is the Wuxia, another one is WOX11, also a Wuxia homolog. So basically, Wuxia activation works for poplar regeneration. And as you know, this morphogenic factor gene is very hot recently, just because of the potential to promote tissue culture, right? So I think there's a lot of room for discovery. If you figure out, which gene you activate in, let's say, other polyploid crop. So then, yeah, I think the same principle holds. Then you can promote tissue culture while also, because the promotion of tissue culture is through the expression of the CRISPR activators, which means CRISPR is expressed highly. I can imagine the genome editing will be also high in those plants compared to regular CRISPR-Cas construct. So yeah.
Okay, thank you. Okay, so any other questions from the audience? Please.
Yes, I have.
Yeah, go ahead.
Okay, Dr. Chi, thank you very much for your interesting talk.
Hi.
So my name is Junxian He. I'm Professor Lam's colleague. So I have 2 questions for you.
Sure. Nice to meet you. Yeah, sure. Go ahead.
First, the question is because after you do the genome editing, you need to do selection, right? To select the positive plants.
Right.
So in your lab, how do you do this selection? It looks like you are very efficient, right? Can you tell us your method for do the genotyping for positive plants?
Oh yeah, okay. So yeah, I mean, all the study I showed you today is all through tDNA-based transformation, nothing fancy, just like conventional way of making genetic transformation, GMO, right? So yes, with CRISPR-Cas expression. And your question is how we select a genome editing line. We have, so you can, the most practical approach we use is deep sequencing, amplicon sequencing. For example, we have, in fact, what we are using is mostly one of the called a high-tom method developed by a group in China. If you search high-tom, but that is really just one of the methodology documenting using amplicon sequencing to do genotyping. For example, if you do we do a multiplexed genome editing all the time. Sometimes we have, let's say, 8 target sites in the genome in the rice being simultaneously edited, right? So and then you have 50 plants, individual transgenic plants, right? So you want to know, which plant, how many of them, whatever edit genotyping. So yeah, we really use that high-tom. We can barcode. We PCR amplify each of the target gene in each plant, And then we have secondary barcode primer, which is already a universal barcode primer in the lab. And we use that because the first round of PCR already have barcode, and then allow the annealing of the second set of PCR primer. And that, the first round of PCR primer is of course varied based on which gene you're amplifying. But the second round of barcode PCR primer is universal. You can just use them routinely because barcoding, you can do like 96 well PCR and then multiple 96 well PCR. And eventually, I don't know how many, because I'm not the guy doing the experiment in the lab. So, but you can basically pull the half plate or full plate together to send as a single sequencing. Like in our case, a single amplicon sample sequencing is really maybe $50. And then depending on how you do that, a little bit more can be $75, but sometimes just $50. And then, but that can sequencing many samples, and you get to like tens of thousands of reads, like 50,000 reads, let's say, right? So for each target gene, you just need maybe 1,000 read, okay, enough to figure out, what the mutation, right? Because if it's heterozygous, ideally 1,000 reader giving 500 reader going to have that mutation. You know what I mean? It is very, very, I think, sensitive methodology to detect all the edits at the target site. So this is what we do in our lab.
Okay, thank you.
Sure.
So the second question is, you have talked about this combo technology. Looks very powerful, right? But still, this technology looks like based on gene-specific genome editing, right? So if we want to edit many genes at a time, probably still, I think it's challenging, right? Because you have to design a guide RNA for each gene, right? So is there any way that can make it simple? For example, like the TDA activation tagging, right? You use one vector, you can put it into plants and they can activate them, many genes randomly. Can we do this by CRISPR technology?
Oh, well, yes or no. So if you want to activate the gene randomly,
yeah,
I really think you just should use the the transcription
conventional way,
conventional tagging system. Okay, so because that is allow you to do random activating of gene. Of course, if the genome is too big, you just waste too much resources to do that. It's not practical. But that's why activation tagging only used in smaller genome, like poplar, like Arabidopsis, rice, things like that. But anyway, I think CRISPR, the beauty of CRISPR is really its specific, its specificity. It can target a specific gene, right? So like I showed you, I can activate a specific gene, one gene at a time or two genes at a time, and I also showed you 5, 6, 7 genes at a time with the metabolic pathway genes, right? So you can imagine if you have let's say, for example, my lab, we are interested in activating all the transcription factors. Right, because I mean, if you look at Arabidopsis rice, you have about 1,600 transcription factors, and we actually have we are making the library of the transcription factors, and we design 3 guide RNA per gene, which still you only end up 5,000 guide RNA for all the whole genome. Okay, so and then we clone them as a single guide RNA because you just mix them together, you synthesize the library together, you clone them together, everything just like treating one guide actually, and then. That is maybe the idea close to what you are thinking. Then you can really transform them to do the screening. But I would argue this is much better than the transcription tagging system because you can define which gene you want to activate rather than randomly doing that in the genome. So anyway, yeah.
Okay, thank you.
Sure.
Thank you, Jin-san. Any other questions from the audience, from the students?
Students are too shy. Please ask any question. There's no stupid question, I can tell you. That's always a good question. Just anything. I go very fast, and certainly something you don't understand, feel free to raise your hand and ask me. So I'll be happy to.
Okay, maybe I can ask one more question. For your combo targeting, okay, so why you always try to edit these multiple genes? For example, the one with the FT, right? So FT causes the early flowering, right? Why you also targeted the other 2 genes?
Oh, okay. So maybe I didn't explain very if you don't mind, maybe students can also benefit. Let me go back to that slide.
Okay.
You can see the slide, right? So let's just do
Yes.
this way.
Mm.
So if we wanna do genome editing, right? So I mean, I'm just using Agrobiotechnology as an example here. We showed in this paper. So Agrobiotechnology is easy. You just did floral dipping, right? You screen, and let's say hygromycin, then you have to screen in petri dish in this case, transgenic T1 generation plants, right? then you will be doing genotyping as we just discussed, right? So then you get seeds, and then you go to the next generation T2, and you will get the genome edited and also transgene free plants.
Right.
Right. This is what everybody's doing, genome editing, right? This is the process you're doing, right? So with combo, so it is not random. So FT is actually a secondary thing because what we really care is genome editing, right? So let's say you want to editing. In my case, I showed you is 2 genes. So let me show this way, larger. So in this case, we have I showed you is like well, in this case, it's actually base editing now. The AAS and ACC2, the modification of either gene can result in the sort of the herbicide resistance to different herbicide chemicals. Anyway, this is I showed you the resistance to the AAS gene. But you know what? This is just proof concept. So I don't want to show you more. I can just show 1 gene, but I show 2. Just remind the people CRISPR-Cas can do a multiplexed genome editing. But what I want to say is when you apply CRISPR, you are really interested in applying that to your targeting your gene of interest, like here. FT is not your gene of interest at all. Okay, so but why do FT? because FT allow you to get early flowering. I mean, you're getting seeds much faster. You're saving you a lot of time. That's whole idea about the genetic basis of speed breeding, and also FT activation because of getting you early flowering plants. And we found these early flowering plants are really enriching high efficiency genome editing compared to the large dynamic range of CRISPR construct typically you see here. So which means early flowering plants representing more robust genome editing compared to you don't have that FT activation, just normal CRISPR-Cas vector. Okay, so that is second advantage. You are enriching because when you do genotyping of your individual transgenic plants, you better if you know you are selecting plants that are highly with higher efficiency genome editing than average, you'll be happy. You know what I mean? Everybody will be happy. So otherwise, like I said, how can you do something efficient? Because I have to screen so many plants. It's just so much effort, so much wasteful of my money. So you really, this is really basically both this approach and later I showed you the rice hormone-free genome editing is really themselves by itself is really a genome editing technology improvement. Okay, they are really improved technology by itself. Okay, so I didn't really spend much time, but I hope you understand. And then the third advantage is later on you still have to, like I said, I have to genotype. I showed you we do the kind of deep sequencing genotyping, but here you have no idea. You just have to genotype everything here. But now here you just genotype the plants which are not early flowering, just normal like wild type, right? Because you are saving 75% of chance if we assume transgene insertion is 1 copy and integration, and which means based on Mendelian segregation, 25%, 1/4 are wild type look like without transgene. But now just looking at the plants, this plant is without transgene, or these plants actually have transgene. so just only focus on it. We don't do the NHPC at all. Just focus on these plants. So it's actually you only need to genotype 25% of the total population as you normally have to genotype. And then you can get to
So
I think that's a lot of
the gene of interest. I see. Okay.
Yeah. So
thank you.
Yeah, sure. That's why we actually developed this system at first place. So
okay. Thank you.
Sure.
Okay. Any other questions? Okay, then I asked my question now. so I asked my first question about the off-site editing. So how about the activation? So will there be off-site activation because you need to reduce the length of the targets, right? So that it is not editing, but instead just attracting activation factor. Will there be any possibility?
Yes, that definitely, like just like PCR primer, you reduce now from 20 to 15. So then that will be having enhanced the chance, the probability of off-target gene activation. But keep in mind, this off-targeting, it may have some off-target binding, right? Because it's reduced length. But in order to have the gene being activated off-target, it's actually tricky because you have to position your guide RNA to a specific window, which is typically from 50 to 200 base pair upstream of transcripting start site TSS. And then if you off-targeting to anywhere else in the random site in the genome or in the gene body somewhere, you are not going to activate the gene, okay? Even though it can bind randomly. So that's really, this second layer of complexity actually reduced off-targeting effect a lot. So finally, I would say as a plant biologist, let's focus achieve on-target editing more than off-target effect. I don't think we have to worry about too much about off-target effect.
Okay.
So,
Okay.
yeah, for the most part, I'm not saying don't. I mean, I'm funded by multiple agencies to do that because I know the USD, they want to know is that off-target. They want to make sure when they write the rule, you know. But frankly, do we really care? We don't care. So yeah.
Okay. All right. So well, last chance. Anyone want to ask question? Students are very quiet.
No problem. I mean, if you later on, if you look at the whatever reference paper or something I mentioned, or my study, which already published to Nature Plant paper this year about CRISPR activation and also CRISPR combo. You have any question, something? Yeah, shoot me email. I may not directly answer you or direct to my poster, but nevertheless, feel free to reach to me, and I'll be happy to help. Okay, so it's my pleasure.
Yes, as I said, if travel is more feasible in the coming years, I would very much like to invite Professor Shi to visit us in person.
Yes.
So that he can talk in small group with our research students. I think they will be more confident to speak out when they have a small group meeting with you. So let's plan for that. So I hope that you can come here in Hong Kong to meet us.
Yeah, I'll be happy to.
Okay,
once the travel become easier, yes, I'll be happy to. Looking forward.
I think next year probably possible.
Yeah.
Okay.
Yeah.
All right.
Yeah.
So yeah, before we end, let me do some advertisements. So today's talk is very exciting. So next month we have another speaker that I think we will have the date. Catherine, what is the date for Dr. Sachiko Isobe for present? I think the date is not correct, right?
Oh, sorry, it's the 15th of October.
Okay, 15th of October, right?
Okay.
So Dr. Isobe has constructed a web for sequence, and this website can be used for both professionals and also laymen. So it's very interesting to see how a Japan group has developed a website for different plant genome. So if you're interested, please join us on August 15th. All right. Okay. So with that, I thank the speaker again, and also thank Caroline Yun who make all the arrangement for us. And hopefully we will meet each other again in subsequent months. So see everybody, and good night for the fans in Hong Kong, right? So, okay, bye bye everybody.
Okay, see you. Bye bye.
Bye bye. Bye bye.